Hello!
Hi there.
Welcome to Microbiology Journal Club, where we nerd out big about all things small.
My name is Danny, and in a previous life, I dropped out for my PhD in microbiology at the University of Chicago, where I was infecting skin agar with MRSA. Nowadays, I'm a fact checker for pharmaceutical advertisers and the president of Biotech Without Borders, a nonprofit based in NYC dedicated and aimed at the public with the tools of biotech.
My name is Faz, and in a previous life, I got my PhD in microbiology from Imperial College London, where, amongst other things, I was making flesh-eating bacteria glow in the dark. And since then, I've also worked as a research integrity specialist, and these days, I'm an editor for an academic journal.
Every week, we meet to talk about microbiology, and today it's our deep dive week where we look at each figure in a paper that we chose last week to learn and criticize the scientific findings. This is a journal club, and we encourage our audience to leave us questions and comments.
Next week will be our news week, where we survey a bunch of different papers and articles and choose one for the next deep dive. So subscribe to make sure you satisfy your microbiology curiosities.
You can follow along with the papers that we discuss on either week in our shared Zotero library linked in the doobly-doo below.
And we want to hear from you, so please use comments or tweet at us using the hashtag MicroTWJC hashtag.
So yeah, we took a break for 1 week and we're back with this deep dive. It's a paper that we chose, I guess now 2 weeks ago,
Yeah.
entitled "Efficient Retroelement-Mediated DNA Writing in Bacteria."
Yes. Now this is an intriguing paper. Firstly, there's like the concept of DNA writing, which I feel like is used in a specific way here. But DNA writing basically is used like to describe the putting together writing of DNA. So I've used it to describe ex vivo chemical synthesis of DNA strands. But here they define it as a genetically encoded device for the targeted editing of DNA in living cells. Um,
Yeah. So I guess like cells always replicate their own DNA. So that's like not that we know we've always exploited that machinery in biotech to create DNA that we want. But I guess what is interesting about the technology they're proposing here, or that they've been developing for some time, and this is the latest installation of their work, is that this is a way to like have a DNA construct that, upon some sort of stimulus, will create a certain edit that's designed, that I guess that's encoded in the way that that circuit was set up.
Yeah, so they call these scribes, I think. Which so I'm gonna put up. So the idea of these are the synthetic cellular recorders integrating biological events. So they come up with a couple of this paper they're gonna be exploring multiple kind of like aspects of this. So the slide below
Mm.
shows you kind of the three main things they want to achieve with this. So the first one is molecular recording, the idea that you can use DNA recombination as an output, and then you can
Hmm.
so if you have a sensor, and then it acts on something within the cell to rewrite a piece of the DNA, and it lets you know
Yes.
that it sensed something, and that stays within the genome. So I mean, it's kind of similar to what the CRISPR system does, I think, but with CRISPR
Yeah.
system,
I mean,
yeah.
it uses the same. I guess it uses the same principles,
Mm,
right? Of like recombination. Is one of the ways,
yeah.
but they did highlight that CRISPR, when it makes its edit, it makes a double-stranded break,
Mm,
and that has the potential for like causing the cell that is undergoing the edit to have some not good effects.
yeah.
Like maybe like sometimes, so like in one case, this is people talk about CRISPR, like it'll make a double-stranded break, and then for whatever reason the repair doesn't happen, so then you get a deletion where you wanted a writing event. Or you could get like a fatality, I guess. Like if everything went wrong and your double-stranded break didn't get repaired properly, you could have the cell die off. So with their system, they're using a not CRISPR, they're using something called a Retron, and that doesn't make a double-stranded break, and so like it avoids these problems.
Yeah, and another thing that CRISPR has, it has to have a in sequence already in the genome in order for it to act. So you
Mm,
have to make so it kind of narrows you the amount of bacteria you can work with, and narrows
right.
somewhat the and there's this concept in I think genome editing of scars. We don't want to leave scars on the genomes of organisms.
Yes.
Um,
Well, the earliest days of genome editing, people use these enzymes called restriction enzymes. I mean, I guess people still use
yeah,
them, but those enzymes they cut at specific sequences, and so you essentially make like compatible ends, sticky ends is what they called
yeah,
them. But when you reconstitute things, they'll always have the sticky ends still at the end of where whatever you cut out and whatever you paste in. So that's considered the scar, and it's just people want to when they make an edit. I guess it would be nice to have like no trace of that edit,
yeah.
because that would let like a more cleaner. You're like removing variables, right? Especially if you're doing some sort of experimental design, you're removing variables that could confound whatever your results are.
Yeah, if you want to like splice two sequences together in a certain way, having that scar can interrupt those sequences and cause some problems in the way it's read out or how or the ultimate protein that's
Hmm.
made out of it. It's
Right.
so it's there's a lot of different reasons for why that. That's why they're quite interested in having. So I think this idea of a molecular like recorder for what's happened to a cell is very interesting. Um,
Yeah.
they also like talk.
Um, oh,
Sorry.
go ahead.
Uh, I was about to move on to the next thing, so I don't want to like override your
Oh
point.
no, yeah. I just wanted to touch again on that cis
Yeah.
element part. Because I'm not sure if we like necessarily have spoken so deeply about CRISPR in the past, but everyone touts it as like you can edit anything, right? Like all you need to do is like have this sequence. All you have to do is uh, find the matching sequence. You make this guide RNA, and then the guide RNA sits down on that part in the genome, and then it directs the action of the protein to do the snipping, and then there's some repair event that happens. But I guess if you haven't made your own guide RNA before, you may not know that like those guide RNA programs, they do require there's some parameters. The guide RNAs can't sit anywhere on the sequence,
Mm.
but within a gene, which is like maybe like 100 sequences long or whatever, or that's a very short gene, 100 base pairs, right? Usually genes are in like the thousands of base pairs. You're usually likely to find one of these, like they call them PAMs, I think,
Yeah.
one of these elements. And we actually have I think on the docket we had like a paper we were considering that they were talking about, oh, can we like mutate these PAMs, right? Can we
Yeah.
mutate CRISPR proteins to use different PAMs? Because it is a limitation of targeting. And especially in an application like they're describing here, where they really want to just use DNA as a encoding tool. Having the restriction of directing things against PAMs, like it does, it could make their, I guess their whatever their readout registers, like they're going to use a lot of terms like register, meaning like the memory of the DNA. It could make that construction more complicated.
Yeah. And the second thing, so moving on to highly efficient Scala, so we've mentioned that. Also, editing in situ within bacterial communities, and we have and also spatially and or temporarily regulated diversity generation. So the idea that you can actually dictate when evolution can happen in a certain area. So a new way of
Yeah.
mutating E. coli.
Yeah. And essentially what they've done is like this Retron, again, we've touched on it before. It's a different type of editing procedure, I guess. It uses a different set of proteins and a different mechanism, and people are exploring all the different ways that it can help us achieve some of these interesting research and engineering goals.
Yeah. So yeah,
Sorry, that's all from me
yeah,
from
that's
that part.
excellent. Okay, so moving on to Retron. So this
Ah,
is
yes.
a picture of what the Retron is. So it has like three genes attached to it. There's the MSR gene. There's a reverse transcriptase, which is just in green dictate, and that reverse transcribes the DNA sequence. I think it's the RNA. Sorry, it reverse transcribes its sequence into like well, firstly, sorry, I'm getting confused. Firstly, it transcribes into RNA, and then
Yep.
it transcribes a reverse transcriptase, which uses the RNA to prime itself to create a single-stranded DNA as part of
Mhm.
it. So it's kind of got this mix of RNA, which is the MSR3, and then it's got this single-stranded DNA on it. That's usually the
Yeah.
MSD gene. And what they can do is they can take this MSD gene and they can add in a gene of interest. So you've got
Hmm.
so in this case they've got like a kanamycin gene, um, and they can tie this up with another thing called a beta recombinase. My
Hmm.
animation didn't go. But anyway, so to do this thing called homologous recombination. So you get your gene. You got kanamycin on this ssDNA. You got kanamycin in the genome. The kanamycin
Hmm.
in the genome in this case has been the green bit is indicating that it's off because I got confused when I was writing this. I had very little sleep. But yeah, we've got this beta recombinase that appears, and its job is to and when it sees like two strands that are very close together, they're very similar. They got region of homology,
Hmm.
and so it can splice those together and effectively swap those genes around. So you can get
Yeah.
the region of homology on the retron swapped over onto the genome.
Yeah. So I guess what's really so it's these two. I think the two elements that you've mentioned are the important ingredients into this system in which they can make single-stranded DNA from something encoded in the genome. And that's done through this mechanism that you just described of reverse transcription. And then the second part is getting that single-stranded to recombine into some site. And so with those two elements, so I guess one of them is the retron is like this strange hybrid structure. Yeah, I think it's quite interesting to think that like something like this exists. I believe that this is from sort of like a bioprospecting
Yeah.
thing that was done in the past, where they were like looking at strange like DNA elements that existed, and this sort of showed up, and they're like, well, this is interesting. This is a way of making single-stranded DNA from the genome. So like, if you think about some of the tools that we've had in our toolbox before as molecular biologists, right? We know that cells make mRNA, or like single-stranded RNA from the genome. We know that some viruses can make double-stranded DNA from the genome, right? Like they can, like retroviruses, right, can do that sort of reaction. You have to go back like pretty far into the history, I guess, of things. People used to make single-stranded DNA from phage, and it'll come back up strangely enough also in this paper, but some of the early like people when people were making oligos to do probing before people had synthesis techniques, they used biology to make it. So they used M13 phage to make oligo probes. Nowadays, people just synthesize oligos, and you can transform oligos directly into a cell, and then use that beta recombinase to recombine it in. But then you have to get those oligos in. So what the retrons give us is this ability to make oligos essentially inside of the cell itself.
Yeah, and uh, yeah, that sums it up, and that's why this technique seems quite powerful because it does
Hmm.
allow for this scarless recombination to happen. Um,
Yeah.
and so this technique is going to be the focus of this paper. So yeah, this basically shows this is a diagram showing exactly like kind of how this scribe system works. So like you mentioned with CRISPR works by doing SNPs, whereas this recombination seems to happen when the bacteria is replicating. So at the replication
Mm-hmm.
fork, which is I mean I'm assuming I'm hoping our viewers have done some level of biology so they know what.
Yeah, well, but this is what we were, yeah, this ties into what we were saying earlier in that um, it doesn't actually produce any damage,
Mm,
right? Like there's no thing that comes in and cuts stuff, cuts the genome in this case. It's during the replication of the bacteria that exposes a single-stranded version of the genome that then the beta recombinase can act on.
yeah, uh, exactly. And so uh, yep, I've got my little diagram of beta recombinase in there. Um,
A technical drawing.
yeah, very technical. I put a lot of effort into these. um, yeah, and the thing about the scribe system is that they've worked on it previously, and it wasn't necessarily very as efficient as they want it to be in order to do what they want. So they have to like think about the things that might be causing because cells in general don't like to have their DNA messed with. That's just a general rule. They hate it when
Yep.
when you add foreign DNA, and they've got these different like mechanisms, the enzymes that are there to destroy sort of DNA. So I mean, you've got your
Hmm.
recombinase, but you also got these nucleases that chew up the single-stranded DNA that we've talked about is really important to have this process work. So what do we do about these? Well, the thing we do is we find the genes that create these nucleases and knock them out. And once you knock those genes out, you knock out those nucleases and you remove an obstacle to this happening. So this is what they did.
Hmm.
They created some E. coli where they knocked out certain genes that they thought might be certain nucleases or uh, genes that they thought might be antagonizing this process. And
Hmm.
they found that the ones were that did the most recombination when they were knocked out were the Zon A and Rec J. Um,
Yeah.
so
Which are those? These are two like exonucleases, right?
yes, they chew
They
from the outside
degrade
in.
yeah, they degrade DNA from the ends.
Yeah, I believe like Mates might just chop them into slightly smaller pieces, but
Hmm.
and I think XZA might do the same thing. Let me double check my notes quickly. Uh,
Um, and while you're doing that, I'll just say for those who are watching, these types of mechanisms, right? We've talked about phage before. Like a lot of, I feel like the bacterial life defense mechanisms is trying to defend from phage. A lot of mechanisms are in place for that. And phage have DNA genomes, so mechanisms exist to prevent these types of events happening. Because you could imagine in evolution, right, it might be favorable for a phage to, I mean, this does happen, right? Phage have sequences that are homologous to sequences inside of the genome of bacteria. I think we've seen examples of that in some of our phage coverage, and they'll use that to recombine into the genome to have that lysogenic phase, right? Or maybe they'll recombine into the genome for some other nefarious purpose, like we have yet to discover. And so bacteria have all these mechanisms to prevent these types of things, and but as engineers, we want to exploit those mechanisms, right? We're discovering things, and we're like, oh, we want to use this phage-like thing, this self-replicating thing, to make changes that we want. But yeah, sometimes you have to do that in a different background, which is actually really annoying if you think about it. Like to think about, oh, I want to engineer this thing, but actually I can only do it in a very specific genetic background. Um, but that happens a
yeah,
lot. This is like a typical way of approaching like a biological engineering problem, is that like you kind of remove the barriers and then maybe you can figure out a way to get there in a different way. And that could open up the technology to other uses. And I think we'll see that in this paper.
yeah. I mean, speaking of that, yeah. So I've looked at my notes. So they're all exonucleases except for MUT-S, which is a
Mhm.
mismatch repair system. So, like you
Ah,
said, when
yes.
there's a mismatch, when the phage like enters or any foreign DNA, it checks it and it's supposed to correct point mutations of DNA insertions.
Hmm.
But I think there is a limit to the length it can deal with,
Hmm.
and I think that these insertions are kind of just out the range of where what. So it's not looking like it's doing that much
Yeah.
to
Yeah.
decrease
So
it.
mismatch repair, the reason why mismatch repair would potentially disrupt the system, which it doesn't seem like it does, but the reason why they thought that is, maybe if we go back one or the slide where we're just
Yep.
seeing that primer land, or that single-stranded piece land on the genome. Yeah, this is, oh yeah, this
Yeah.
one's the best. Yeah, so that, right, that single-stranded DNA, which is getting grabbed by the Bayer recombinase, it sits down on the genome right afterwards, right, on the CANR. And where that
Yeah.
star is, that's the mismatch, right? Because we've made it, the idea is in this technology that red piece is something that we control. And we have the mutation or the desired sequence in it. When it sits down on that surface, that's a potential for the mismatch repair to come in and say, oh, good, we don't want to make this in this way. Let's like cut it off and make sure that we just follow the regular template, not this weird one. But I guess, you don't know what will happen. This is sort of a big, right, inside of these cells, it's a big bag of proteins all doing their jobs. And we don't really know the relative contribution to the different elements, especially when we're throwing like this huge wrench in the mix to try to get us to get a desired result. And so they tried to knock out a whole bunch of different potential control mechanisms here. I guess mut S is not that important in this particular process.
Yeah, apparently it can't handle base mismatches longer than 3 base pairs. So if you're inserting a whole gene, it's just too much for it to deal with. Uh,
But these sequences, I don't recall now. Do you remember how long these sequences are? These single-stranded
I think
sequences?
like maybe 6 or 7 nucleotides long.
Okay, I think actually they did. There was a figure where they looked at like the length of it, the length of homology versus the efficiency.
Yeah, well, so
It was
80.
under 100, I
Yeah,
think.
under 100.
Yeah.
Well, yeah. Uh, and so okay,
Okay.
in this case, so we're looking at, yeah, this is more about looking about that in more detail. I think same data. Um,
Hmm.
but again, focusing on the different enzymes. Uh,
Yeah, and they're using in this case they're using the IPTG to turn on the retron, right?
yeah,
Hmm. So like the creation of the retron is under control of this IPTG model. For people who know protein expression, right, that's a really common way that we control expression. It's like a chemical switch that we can turn on and off.
yeah. And the main issue here is like you said, we don't want to use a strain with a specific background, so we don't want to have to be tied to this one E. coli strain that has all its knockouts. So they decided
Hmm.
to include a way to knock out these genes in the system by using CRISPR. So we've got these genes that produce exonucleases, but now we've got CRISPR with these guide nucleases that are targeting these genes. So
Hmm.
so that means that you're not just introducing this kind of high scribe to this scribe system. You're also introducing all these CRISPR systems that are designed to defend it against these exonucleases. So they basically smack the genome and take out the exonucleases, making it,
Yeah.
yeah,
And they're using like CRISPR I, like
yeah,
interfering CRISPR. So like, um, I guess this is really similar to silencing RNA,
yeah,
actually, right? Like this essentially is just saying that like the CRISPR system makes something, and then for every, yeah, I think it is for all the mRNA, right? It binds to it and then stops it, or it sits down on the genome and it blocks the
yeah.
transcription, I think.
It
Yeah,
basically, yeah, it sits on the genome and blocks transcription. Uh,
yeah.
yeah,
Yeah, so it doesn't it doesn't deplete it depletes the messenger RNA. That's the outcome. That's why it said it's similar to siRNA,
yeah,
but it does that by taking up space on the genome where you would normally get the mRNA to come off.
yeah,
But that's nice because you just said it, but I'll
yep,
repeat it because like that's the important point. Like it's nice because you don't have to make a mutation of your parental strain, right? It takes time to make a mutant. Mutants are also like susceptible, right? Like let's say we want to take we want to take our cells and put them out in real life environments where they might find dangers like phage. If we were limited to only using the system in these kind of like nerfed backgrounds, then we may be limited in the types of investigations we can do because they may, get hurt out there and not be able to actually survive long enough for us to even see the changes, or they might like get infected by something, right? And then we don't even see the changes that we're asking the bacteria to write in itself.
yeah. So yeah, next picture basically describes how that system works and shows the graph
Hmm.
of it working. Of uh, they adding in the CRISPR and they've got the two controls, one with out guide RNA, one with guide RNA for RecJ and Zone A, which are the two exonucleases we want to get rid of. And yeah, once you
Hmm.
have, and they're both under, so the CRISPR is under the ATC, so I think it's acetotetracycline or something like that. I
Yep.
need to check my notes for the actual proper name.
Yeah, it's something like that. I know it is tetracycline, so it's like from tet-inducible promoters.
Yes,
That's a antibiotic, but it gets like this the whatever the A stands for is added onto it
anhydro,
actually, and
anhydro,
hydro.
anhydrotetracycline. Um, so yeah, once they induce, so they can add IPTG and anhydrotetracycline, and with both of them, you get much more recombination. It's quite interesting how they're using recombination like frequency as the output for their sensor. I think
Mhm.
that's something that's going to be we're going to be having a lot in this paper. Um,
Yeah. Well, they do it. It's actually it's quite it makes this a really nice assay. It makes it a very easy assay, right? Because their parental strain isn't resistant to kanamycin. So if recombination happens, the strain should become resistant to kanamycin and then grow on kanamycin plates.
right.
So all they have to do is count how many colonies, right? Is telling them the efficiency of what's going on.
Yeah. Oh, colony
Yeah.
counting, the most favorite part of a microbiologist's day. I mean,
Right, right. Well, I mean, it's like, yeah, I guess maybe like for people who are working at the bench, like, okay, you're counting colonies all day, but really, it's also that's the power of microbiology, right? The fact that single cells give rise to something that we can see with our eyeballs, and then that is indicative to rates of a reaction happening inside of those cells. That's one of the reasons I love microbiology is colony counting. But I guess it's also the bane of my existence sometimes if I'm counting tons of plates day in day out.
oh yeah, using the felt tip strategy of dotting every single colony until your plate turns black. I mean, oh. Vibes.
All right.
Let's go. Oh yeah, and now they were looking at the recombination frequency based on the length of region of homology. So the region of homology is a bit that's going to be ahead of the insert usually. So I was always kind of
Yes.
taught the longer the better. But
Yes.
uh, it,
There's a sweet spot
yeah,
here.
it seems like 35.
Uh,
Yeah,
and that's probably based on right, like that's based on some property of the beta recombinase and like what during the replication during a replication event, right? Sort of like what will cells tolerate for a single-stranded piece of DNA, like sort of jamming up the works of regular replication and saying, actually, no, this is the proper template. That's yeah, that's what we're seeing here.
yeah. It's a nice thing to establish so that you can design your inserts better. And
Hmm.
also, they decided to improve the beta recombination expression by adding a stronger ribosomal binding site. I mean, it's a subtle thing, but these things do actually can make quite a big difference. Um,
Yeah,
so that's
and to me, this also seeing this figure, it gives me that sense of like how we're really in an this is like this is genetic engineering, right? We're seeing that fusion, the synthetic biology and genetic engineering. It's a very engineering approach to things, right? Like they've created this object that now they want to up some of its properties, right? How well does it work? How frequently does it work? Because by improving these properties, they hope to improve the types of applications or expand the types of applications they can be used for. As I mentioned earlier, we had a way to add oligos into cells, right, and get this type of single strand of recombination to happen. So we've had some ability like this. It's that the fact that we can encode them on these plasmids essentially, right, and induce them whenever we want, instead of having to transform cells over and over again. That is giving the advantage and the application space for this particular technology.
yeah, for sure. I mean, because just single cell DNA isn't sorry, single stranded DNA just isn't
Mhm.
sturdy. It's not very stable. But having
Yeah.
it on a plasmid and being able having that sit in the cells and us being able to control when it does the recombination, that's
Hmm.
very powerful. And yeah,
Yeah.
think
And so by playing around with the construct they make, we can get even better profiles. We can make more of that single-stranded RNA, right? Or we can increase. In this case, they're trying to get more beta recombinase so that it's more likely that that beta recombinase is going to do its recombination action.
yeah. I mean, this paper basically has like the main. This is a section where they try to make their model as good as possible. May they do as much recombination.
Hmm.
So they've taken out the exonucleases. They've added more ribosomal. They've added better ribosomal binding site to get more recombination happening. So that's got and next they're going to be testing how subtle they can be. So making synonymous changes.
Hmm.
For those of you at home who don't know what synonymous changes are, they're changes to the genome that don't change the actual output of the the
Hmm.
phenotype. Um, so like for lots of different proteins, they have multiple different like sequences that code for the same protein, just switching in and out between
Yep.
them. And this so you
Hmm.
wouldn't actually be able to. So this is one place where cell counting won't help you, because the phenotype will be the same no matter what. So you'd have to resort
Yes.
to other techniques, such as genomics,
Sequencing.
sequencing. Yes.
Hmm.
So it,
But we've seen that nowadays you can sequence anything and everything. So this really is on the back of having the ability having these high throughput sequencing machines where you can take your population of cells, blend them all up, extract the DNA, and sequence a particular region.
yeah, and I think this is like a good time for them to demonstrate how subtle they're, how they can do this scarless genome editing, because this is the kind of thing that wouldn't be possible with, say, a proper restriction enzyme system or some. So,
Yes.
and here they do their sequencing, and their output is essentially to find areas where the code isn't very stable, because that would indicate that
Hmm.
there is some diversity there that indicate this anonymous
Yes.
mutation.
And so it worked,
Yeah, it
is what they're
works.
showing us here, right?
That's the main thing.
Yeah.
Um, and so they look at the
Oh, actually, wait. Can you go back? You can also see that. Oh no, to the
yeah.
sequencing
Sorry.
information. You can also see that when they have a 2-day cycle, right? They like transform it with the retron construct. They add the inducer or whatever. So now there's like single-stranded DNA. In that first day, it you can see the sequence. There's like uncertainty, right? Those bars are like how certain the call is for the read. And so you can see like, oh, it's kind of uncertain. It means there's probably a mixture of colonies in this on this plate, right?
Right.
Or cells, I guess. There's a mixture of cells. Some of them have been edited, some of them haven't, but the consensus seems like it is the edited form. But by day 2, after they restreak them, so they take those cells and they streak them out again, you can see the confidence is better for the things that are grown. And again, this helps to highlight that fact that it's these changes happen during growth. So it's possible that like not every cell is going to divide right away, or like have everything all repaired up by or have the event even happening. But once you wait a day, right, things have resolved themselves, and you get like more of this edit yeah,
Yeah.
in the population.
I mean, that's the strength of having a plasmid whose function is to be genome editing all the time. Because a lot of these, like with electroporation, we put something in and we hope it'll happen once. Whereas with a plasmid, it's
Yep.
sitting in the cell, it's constantly pumping out these RNAs and
Yes,
ssDNAs.
that's right.
So
As long as they're receiving the signal, which is the IPTG in this case, right? That they'll be making that single-stranded RNA and editing stuff to become more well that we want. Yeah.
yeah, totally. I mean, I remember I was doing gene editing with electroporation for like suicide plasmids to go into a cell and just electrorating for months and months on end for something that's a very low probability to happen. And then they can just put their plasmid like once or twice, and the plasmid will do all that work for them. And that's quite.
Right. Right.
Uh,
Yeah, and it's not that you can't get rare things to happen, right? Like I've said before, people have electroporated oligos into things, right? Like people even just electroporated whole CRISPR systems, not on the plasmid, into
yeah,
things, right? And as long as you have like a good selection mechanism, you can always get out what you want. But the strength of this technology is the fact that like you can always turn it on. You can make sure that like through every growth cycle, if the cells have experienced the signal, the change is going to be happening.
yeah.
Okay.
And the next page basically shows what you've just said, but in graph form, where
Mm.
yeah, in one 60% have the allele like changed over, and then by day 2 even more. It's really
Yep.
getting the plasmid, the genes to do the work for us because they've got this really good genetic
Yeah.
circuit to happen.
Yeah. And they have a good. This is a good pictorial representation of that, right?
Right.
Yeah. That like through every generation, as long as that signal is present, right, that retron is going to be pumping out those single-stranded and getting more of the population to transform or be mutated in this way.
Yeah. And even if the transformation doesn't happen, the plasmid is passed down in the generation, so it can still happen a generation later. Uh,
Yeah.
so yeah. So this essentially shows the same thing kind of happening. Yeah. A lot of the
Yeah, what did they do? This is a different
yeah
background.
a,
Yeah.
a, a different
So they were using
galactose K, I think. which is
No, what would I want to see what cell background were they using? Yeah, right. They were using delta exon delta rec J, right? So those
right. Yeah,
those are the ones where they took out the two nucleases, right? So remember, like that's like the okay, like that's fine, but like we're not always going to be working in that background because it has a higher propensity for other mutations that like might mess things up. So they just wanted to see like what are the rates like in a background that doesn't have those things.
right. Yeah, and I think
Right. This one is what
so.
does this one say? Yeah, this is exo minus.
Yeah, they get rid of the exonucleases, so this one doesn't have the CRISPR attached to it. And this, instead of looking at synonymous mutation, this is looking at the an on-off mutation. So when it has the mutation, you get pink colonies, and if you don't have it, you don't get those pink colonies. And
Oh, so actually, wait. Exo in this case is exon, and
oh,
or is
exonucleases.
the
So
yeah. So it is this
yeah,
background.
it is this background. So
It is the
so
same.
it's the
Oh,
same.
okay, that's my mistake. Sorry.
It's all right. The terminologies of these things can get on my nerves sometimes as well. But yeah,
Yeah.
it's again demonstrating that same thing, looking at the sequencing this time. So rather than just looking at the frequency of like, oh, they actually delved in and showed the same things happening, which is quite
Mhm.
yeah. So it's kind of like adding more evidence, adding more to this
Yeah.
concept. And oh, the next one is quite interesting. So CRISPR counter selection. Um,
Hmm.
so the great thing about CRISPR counter selection is this is essentially getting your CRISPR and having it target the unchanged gene. So you put this
Yes.
plasmid in, hoping that it'll change your gene, but then you put this CRISPR in. So if this damn bacteria doesn't take your gene, we're gonna have a CRISPR to spite it and destroy it in its
Yes.
presence.
Yeah. Yeah, and this type of negative or counter selection, it's also like an old concept, right? In bacterial genetics, we would have these genes that sort of like placeholder genes inside of the parental strain, and so when we turn if we add an inducer, that placeholder would be toxic in some ways, right? And then we would turn on a toxin gene, right? That would kill the cell. But here, I guess what's wonderful with CRISPR technology is that we can make any gene the toxic gene, right? Because just by having this sequence, CRISPR is going to make a double-stranded break that's unrepairable in that sequence. And so, yeah, so that's like, uh, I guess this is maybe how far we've come, right, in genetic engineering, that we're not limited anymore to like thinking about counter selection as I need to express this lethal gene, but rather we can say like we need to just be able to target a lethal CRISPR SNP, right, in the right location, and then that's going to mean that there's a selection pressure, right, inside of the genome that we've created inside of these engineered cells. If they're not getting edited properly, they're gonna die.
Yeah.
Hmm.
So yeah, that this diagram basically illustrates what you just said. I should have been a bit quicker on the click up, but yeah,
Oh yeah, we're
yeah,
good.
we're good. And yeah, when you activate this, suddenly the allele frequency that take this up is much higher. So yeah,
Yeah.
this big red line is just like 100%. So it's pretty good. Yeah, this this like kind of method of doing genome is essentially engineering something to do the genome editing for you, which is quite good.
Hmm. Hmm.
Uh,
Sort of all in one step, I guess, is
yeah,
the other thing, right? All you have to do is add these inducers. You don't have to do a restreak or anything like that, right? Like it's all just like this is what it looks like on the plate.
yeah. And now we've got like phage mid delivery of CRISPR I high scribe. So they've got like they managed to pack these kind of constructs all into a phage, um,
Hmm.
and that's interesting because then you can just put it into a community. You don't need to because usually with like these you have to put a pure culture, but then they have this idea of adding it to a community of bacteria that are mixed, and so some of them will be susceptible to this phage and will take up the gene. Um,
I I think yeah, it's not even. I don't think it's even about. Oh, it is about susceptibility, is it?
I don't. I,
Uh,
I,
I thought, oh, I see. No, I see. It's because in the community, they're targeting a specific bacteria with this phage in this instance, that's how they're imagining the application.
Yeah. So in this case, they take a bacterial community from I think some mouse species
Yeah.
that they kind of extract bacteria from, and they mix it with everything else,
Yep.
uh, just to get like a diverse community. And then they see whether they can manipulate the genomes of this specific species within that
Within
community.
that community. Yep.
Yeah, and they find that they can do it. Again, something you wouldn't be able to do with like chemical like versions
Yeah.
of this kind
Yeah,
of
because if
process.
if you were doing electroporation on this, you would get that same low chance of putting the plasmid in, or yeah, putting the construct into all the cells. But because they can package it in something that has specificity for a specific cell, that gives them this ability.
Yeah. Again, quite a great good thing to have. So I mean, in terms of like thinking of if you're trying to study a bacteria in a community and you have a phage that can target that bacteria, suddenly you have a system to if you've got understand how the phage works, you have this new system that you can use to target and genetically engineer
Yep.
this community, which is
So we haven't covered too many like microbiota papers, but like you could imagine in this way, like you could just feed the mouse these phage maybe, or like I guess maybe
yeah,
enema the mice with the phage, and hope that like that mixture interacts with their community, and now like now you can have some sort of reporter that you're interested in making genome edits in the bacteria in the gut of the mouse, right, in its context that it's normally in.
Yeah, I mean, this is going to be quite an interesting thing for people who are interested in phage therapy, because I'd be interested in what they come up with for this, if this, because this could be really interesting technology to apply to that.
Oh yeah, like I mean, I guess so. It wasn't. That seems so like yeah, like
I
because you'd be directing like very specific toxicity to certain cells maybe only when they come into certain circumstances, right? The power of this is that we have control over the construct, right?
yeah,
So IPTG is what we've been using in the past figures, right, as the way of turning on the retron. But you can imagine it can be turned on by a variety of different things.
yeah,
So in the therapeutic realm, maybe you don't always want to kill a certain bacteria. You only want to kill them when they've experienced a certain type of environment. Well, that is something that
yeah,
happens, right? There are many bacteria that they only turn on their toxin in a certain circumstance, right? And otherwise, they're regular community members of your microbiota. So you don't want to blast
yeah.
everything away necessarily. Maybe this technology could be used for that.
Yeah,
um,
in a previous paper they use AHL, so the bacterial pheromones that are produced by some species as
mhm.
a trigger for this kind of system.
Yep.
So if you imagine that you have a system, have a quorum system, and
Yes.
use that to activate this to instead of like turning on virulence, like not turn on virulence, that can actually be quite an interesting and powerful thing to do.
Yeah. Absolutely.
Because I remember like this is okay. This is going to be story time. But like
Okay.
a couple of years ago, I was at a conference. Someone like created toothpaste that had this specific bacterial pheromone in it to make like tooth cavity bacteria not form cavities.
Oh,
Uh,
fascinating.
which is fascinating because I think the theory that was proposed, and this has stuck with me like even 10 years later, is
Hmm.
like we shouldn't just be thinking about killing bacteria, but also like ways to kind of like make peace with them, like to make them less like
Yeah.
virulent.
Yeah,
And
absolutely. And when we as we understand more about pathogenesis, we understand those different ways, right? Those different genetic mechanisms that bacteria are using to turn on their virulence, right? Or regulate because those molecules are also energetically, right? Like it costs energy for bacteria to turn on virulence. And so like I think it's a nice thought to think that they may be nudged in certain ways to not turn it on.
yeah, so going to figure S4A, so now they're going a bit further. So they've designed this construct to now have the ability to be transformed into cells, also
Hmm.
I think biochemical, transduced and
Yes.
conjugated. So they've got the M. So wait. So the RP4 is for phages and M13 is for conjugation. Am I right?
Yes.
Okay.
Um, Yeah, it's whatever the backbone of the plasmid is. They've changed it up, so yeah, it has a conjugate element. I'm not sure which one. No,
But
M13 is from the phage, and RP4 is conjugation.
Right, yeah, I knew I
Yeah,
get that wrong.
yeah, because the phage is called M13.
Yeah, that makes so much more sense. So
Yep.
this construct is good because that means that they can now compare the different efficiencies of these different ways of transferring their construct
Hmm.
into different things.
But it also
Um,
gives them another way of moving this construct between cells, right? Like now you don't have to, in phage, you could also introduce just bacteria that have this plasmid that conjugate with their preferred mate.
yeah, true. So you can actually have this plasmid spread through the community.
Yeah.
That'd be yeah. You're absolutely right. That's really interesting. Um,
Hmm.
Yeah, so I think they first like assessed like chemical transformation in terms of allele frequency. So these are very like low probability ways of transforming cells, I think. Uh,
Yes. Yeah.
so the first test was chemical transformation. The second one is let me pull up conjugation. So
Hmm.
conjugation is basically bacterial mating where they swap plasmids around, and that's
Hmm.
how genes transfer in communities, and they found that the recombination frequency was actually really high with this, even in the community, even when they.
Yeah. And I find this is interesting because this is going to set up, I think, for their next figures as
Hmm.
well. But conjugation requires these bacterial cells to be close to each other. They have to be like spatially in the same area. So this is also like now if we introduce, so I mean that was true for the virus as well, but we weren't really studying the virus per se,
Hmm.
right? Like the virus was just a way to get things in. But here, if we're using the same bacteria, you know, this bacteria that lives in this environment, we want to know like does it come into contact with certain things, right? This would be one of the ways in which you could spread this reporter construct, right, throughout a population, but also know that that population is all in contact with each other.
Yeah. Um, so this is the figure where they directly compare like the transformation and the transduction and conjugation.
Transduction.
Um,
Yep.
Transduction seems to be a lot more efficient than conjugation,
Mm-hmm.
but I mean, and that's great, but obviously, like what you've mentioned is very important about
Yep.
how conjugation is dependent on proximity, and that is
Yeah.
going to be the
Yeah.
focus of I think the next few pieces of paper. So the new bits of paper. Um,
Yep.
so here they in the next figure they describe how DNA barcodes can be used to record conjugation events. So
Mm-hmm.
when a bacteria is loves another bacteria and transfers its plasmid over, the recipient barcode and the donor barcode can kind of interface, so you can read both of them and know that a conjugation event has happened between these
Mm-hmm.
specific bacteria.
Right. Yeah. If we think of the mobile elements as containing sort of like unique signals that get written onto the genome, right? The genome is the record of everything that these bacteria have experienced from right using this like Retron writing method. And yeah, it gives us like a really nice report. Or like the way that people would have done this before might be with like fluorescent markers, right? But then like you have to be able to then see like then you have to be able to get those bacteria out and see them, I guess, in some ways to see like they're glowing again. There's no guarantee that like they would have kept that plasmid. Here, they could actually lose these plasmids at some point, right? Like, let's say they go through like a lot of changes, but in the genome, that record will still be there of the things that they've seen.
Right. Yeah.
Yeah.
So yep, they got two different barcodes. One's the recipient barcode, one's the donor barcode. They have like on this
Hmm.
area of the gene they're calling memory tape, and these are which I think they're playing into this idea that this is going to be like acting like memory, like a almost like computing. Um,
Yeah. I mean, they talk about it too that in the sequence, like there's one bit of the sequence is just like the control bit to say like did something get written here. So like it's just like off or on depending on whether something got written, and then the rest of it is that unique identifier that this is the barcode that tells you what was the Retron construct that wrote into the genome.
yeah, and they halve cells and they like put them through this like massive deep sequencer in order to determine all these events.
Hmm.
So yeah, this so in the simplest form, we've got two colonies, each with like a recipient and a donor. Donor has a plasmid, and then in the
Yep.
bits where they interface, so you can pull out the sequence and see whether conjugation events have happened. So
Uh-huh.
what we'd see is like there'd be like non-edited registers and edited registers, I think. Um,
Yes. Yeah, and you can so like that they even show that writing control bit. So like that A, right? Like that doesn't have anything to do with the identity of the barcode. It would just be an A if there was some sort of edit, right? Otherwise it
right.
would stay whatever it is. I'm not sure.
Yeah.
Because this
So,
match up with T. Okay.
but yeah, they've got these like writing controls that essentially the sequencer knows how to find them. So it has like a primers that can amplify up, so you can see when.
Oh, right. That's the other reason, right? Because they do this with an amplification dependent manner.
I think so. I
Yeah.
mean, it could be that they sequence everything and then, but I.
No, I don't think that's how they did it. I think that like it has to have that writing control, otherwise the sequencer won't read it. Like the primers that they're using for reading, like they require that one change.
Right.
Yeah,
Yeah.
because if it hasn't changed, then it won't base pair, and then that's like a floppy end, and
Yeah.
those things don't extend. Those synthesis directions don't go.
Yeah, and now they've got this a line of different colonies with different barcodes, and
Yes.
then they plug them through the sequencer, and you've got this like co-occurrence matrix where you can see. So in red is where you see conjugation events have happened quite frequently,
Hmm.
and then black is where there's been no conjugation events. And based on this, they can try and deduce the structure of like the structural location, the spatial locations of each colony. Uh,
Yeah. Yeah, like so like the idea is that they set up this artificial scenario in B,
mm,
right? They have all their different donor barcode thing, right? Like that they marked all these different bacteria with the different barcodes and they put them in this pattern, right? A spatially isolated pattern. And so they scrape it all off the plate, right? Disregarding all the all the nice dots that they set up, and they sequence the whole thing. From that, they get that information, that heat map that you see, and from that heat map, you can reconstruct what was close to what based on which registers got written to, right? Which barcodes got written to which register, and it pretty much recapitulates the thing that they have going on in B.
yeah, yeah.
Mhm.
So in Figure S5B, they've got even more complicated patterns because they're
Yep.
testing how much kind of detail they can get purely by
Hmm.
sequencing and recording these conjugation events.
Yep.
Um, so
And it works, right? I think
yeah,
they don't know any of the deviations. So I mean, we can even just look at one example. In B, they have a tag. It's D3, TG, TGTA. Where is that?
so yeah,
D3, donor three.
so you can tell it's
Oh, donor.
between two of them. So it's going to be kind of yes, one at the
It's right.
bottom.
It's right here. It's in the middle. Yeah.
Yeah, in the middle. So and you can kind of based on that you can figure out where the other colonies are.
Yeah.
So it's a great way of generating a graph of relatedness. So you can check. I mean, yeah, it's essentially creating like nodes and kind of connections between different colonies. Um,
Hmm.
and then they go a step further, where they so unfortunately this is a very low poly image of this, but it's not showing on the stream at the moment, probably because it looks so terrible. But essentially they basically tag a whole bunch of different cells individually. So imagine if you took all those different tags and then put it just on the cell level. So every single bacteria has a different barcode, and then
Hmm.
mixing them all together and measuring the conjugation events. And based off of that, they could try to build out almost like a relationship map between all the bacteria, which
Hmm.
should tell you something about their actual locations on the.
Yeah, or at least like yeah, well, like what cells they bumped into in that phase. So actually, like one of the things that immediately jumps out to me about this is like not bacteria. I mean, I think bacteria do have these things going on. Like think about like biofilms, right? Like, oh, do bacteria move between different forms in the biofilm? Are there like different sections? Some bacteria, like in the core, are always touching the bacteria on the edge, right? You can sort of parse those things. But I actually think, like, in my mind, the most interesting application, which they haven't demonstrated here, is what if you could do this in like immune cells? um, and you could show like, oh, like this immune cell touched this other cell, right? Like we know that that's a really important feature in immunity, to have like the cells speak together to write to the register these types of events. It is really cool. But again, like, it's a really big system to move this into eukaryotes. But like I sort of smell you going back, like really complicated systems. That being said, we have like a lot of group behaviors as well that we could understand in this way, right? It could be that there's some group of bacteria that have a certain signaling function within a greater community, and we'd be able to see that, like especially in terms of how close they get to different things, different other bacteria. Oh,
Yeah,
this image
definitely.
is having some issues, eh?
Yeah, sorry. We're experiencing some technical issues. So I mean, if you've, so I guess we can try and roll on with the rest of the paper. I guess let me check quickly about what is going on with this because yeah, my share screen is not working at the moment. So
Can you still see it on your computer?
I can't. I think like it's not transferring over Skype at the moment, which is a bit unfortunate.
But you can still see the
Oh
slides
yeah,
on
I
your
can still just
computer.
see the slides on my computer. Um,
I see. So I just have to open up a section of the slides for myself so I can see. I work with the slides here. Okay, and then we can just um,
okay, yeah, we can just try and
to
like
figure that around.
mime things for people then about about how this paper should look.
Yeah, I
Uh,
see. So the next, yeah, you wanted to show us figure figure
Figures,
4C, right? That's the one that we haven't been able to show yet.
yeah,
Yeah.
that's essentially that's essentially describing what we were describing before about like so the technical thing about so essentially doing the process of pulling out each individual cells. Now they've got barcodes, and those barcodes will tell us whether they've got the construct or not. Um,
Mm-hmm.
and it gets plugged into an Illumina system, which can bring out a whole bunch of data that we can look at.
Yeah.
Um,
Yeah. So instead of
yeah,
just the spots on the plates, now we're saying every cell has its own unique donor recipient. You sequence the whole
yeah,
thing. You're showing us next figure S5
S6A.
S6A. And this is what we were
Yeah,
saying, that they do a PCR amplification with the control bit. So that
yeah,
they're only amplifying the sections that get written. they, they're only amplifying the sections of the genome that get written to. um, I guess I guess one of the things that I didn't catch from this is that if they're doing this amplification, then don't they get they somehow have a way of excluding amplification from the retron itself?
yeah. I
Because the
assume that
retron is
they
going to have the control bit in it as well. Oh,
true. Yeah,
maybe they purify the genomes before, away from the plasmid.
yeah. I think that would make sense. Yeah, because otherwise it'd be very difficult for them to find any signal.
Yeah.
Because it,
Well,
but
yeah, they would get all of the. They just amplify the thing that they put in.
yeah, and I think the one things that they did with their sequences, with those C's and A's, I think they could input in they're part of like restriction sites.
Ah, yes,
So
I see.
so that means that if they were edited, then they'd create restriction site areas that could use to chop out. So using that, they could actually pop out all the
Mm,
unedited or registers and just focus on the things that they want to look at, where and
yep.
and send that off to sequencing, which I think streamlines the process a lot.
Yeah, I
Um,
think that's another nice thing in this circumstance, because they can create the register section right, inside of the genome. So you can have it have properties that make it easier to amplify or otherwise.
Yeah, for sure.
Okay, so then we get to figure six, oh, S6b.
Yes. So this is another one that so it would look like a kind of it's a big noodly sequencing output where they essentially use the decrease in quality registers as an output to indicate that there's diversity there and that
Hmm.
registers have been transformed. Um,
Yeah. Yeah, because in this case, this is from the bulk sequence of everything. Things are not going to ever get confident because every cell has a different set of cells. So like it's just going to be confusion in these particular zones.
yeah,
Okay. And then figure 4C is that interaction graph that they end up generating. So
yeah,
similarly to what we saw earlier when they had this like artificial structure, they can see at the pairwise intersection of the donors and the recipients, there's a bunch of hotspots. And from those hotspots, they can reconstruct an interaction map with arrows pointing between all the different barcodes.
yeah, definitely. So I'm doing some tech stuff in the background to try, but I mean, there is one kind of thing that is missing from this that I feel is very necessary, which is um, the question of hold on a second. So, how do we know that these are related in the way that they say they are? I mean, there isn't
Mm-hmm.
any co-registration. So usually, when you're testing, because when you're testing out this kind of technique, you'd want to like. So before, when they're in the original thing, they spot out onto plates, so they knew
Yep.
the structure and they could
Yes.
confirm it. But in the
Yes.
bacterial community, how can they confirm what's happening?
Yeah. Yeah, so I guess you're doubting whether or not like because in the spotting example, they have multiple cells that are in the intersection zone, right? That and we know that they're interacting. Here it's like we have to say that that single cell, right, is meeting another and interacting. But yeah, I guess that might not necessarily be the case. They don't have any independent verification of that. How would they though?
True. Yeah, I think that in that they'd have to have like some kind of other way to. I mean, I guess they'd need a separate reporter
A
of
separate
some sort,
reporter, yeah, and pull
potentially.
out from each of these. There's some. Okay, I'm trying to think like because if the cells grow, they'll hold. If the cells that have seen this history grow up, they will have that edited register will remain in their genome, right? It doesn't matter anymore. They'll have it in their genome. Um,
Okay, right.
hmm. I'm trying to think like whether or not they could be put if separating them and growing them would give you more confidence that these interactions are happening. I don't think it would necessarily. You still have that problem of like having to independently have an independent system to verify it. Um,
Okay, right. Okay, finally got. I'm running PowerPoint off this computer, so things might be a bit slower. But yeah,
okay. Oh, you're gonna throw up the graph that we're looking at.
yep. I'm going to throw up the graph that we're looking at. I just
Slide
need to.
35 of 42.
So slide 35 of 42. Uh, yep, got it up there. Right. So
See.
now you all at home can bask in the glory of this slide. Uh, but yeah, so on like the left side you've got your interaction map, so you see which bacteria are interacting, and then based on that they've generated this really intricate network. Um, so I think this is an advance not just in uh, the sequencing, but also in the data analysis. I think that's quite an important
Mhm.
thing that we've got going here.
But
Um,
I'm trying to think about like your criticism of it that they have no independent verification. Like they had when they did the spatial when isn't the spotting part of that verification?
I feel, yeah, you can say, but the thing
But
that
you're
I'm
worried that
concerned,
like the singles at the single cell level, it's not working in the way that they're saying it is.
so
That's the doubt.
the concern I have is,
Yeah.
well, let's say we have a bacteria that takes up two of these conjugation plasmids what does it look like? What happens when you get two donors and one recipient? Does which one gets recorded?
Mm-hmm.
Um, and so
Two
I'm just
donors
kind of
and one recipient. They already have. Oh, they always have donors and recipients. Yeah, they never have. So I don't understand that because the barcodes, um, how do they? Oh, because they. I don't under, I don't really understand like donor and recipient here. It happened actually in the previous in even in the previous one, right? Like they have ones labeled donor and recipient, but like it doesn't matter who's donor and recipient, right? Like in whoever gets all the plasmid, whoever gets the plasmid with the barcode will have that barcode written into their genome.
right, but I feel like the because one of them is okay. Um, so I feel like
But
kind
they
of
do but they do distinguish in these graphs using blue and green, right? Donor and recipient.
yeah.
And what you just said also it rings true because they never have recipients touching recipients.
Right. They never have like two donors and one recipient. So we never have to ask that question of what happens
No,
when you have like multiple of these systems trying to interact on the same area of
no,
genome, right?
they definitely have multiple donors and into recipients, right? Like that's like if you just look at any one recipient, it'll have multiple donors pointing to it.
Hold on, let me um yeah, they so if we're going back to this that they are, but it's always like the areas of overlap are always between two colonies and never between three.
Oh, I see. Did you go back to S5B?
Yeah, I've gone back
Yeah.
to S5B
S5B.
to yeah.
Yeah, so like this is the I think this is when our technical problems happen. I was trying to walk through one example of it. So in S5B in the first column, right, they have one example where everything's clustered in the top right corner. And if you look at the in the second row, they have D3, which is TGTGTA,
Yep.
right? It's in green. And then
Yep.
that in on the graph below, that correlates with D3 in blue, TGTGA,
Right.
and it has arrows connecting it to the three objects that were touching it, R4, R2, and R5.
Right.
Yeah.
But that's got three. So in terms of like Venn diagram, there's never an overlap between more than two.
Oh, I see what you're saying. Yes, yes, yes.
Yeah.
Hmm.
So it's never in a situation where there are more than two recipients or more than two donors in a single situation.
Right.
So the thing that I'm wondering is what happens when you have say two donors that are fighting to recombine on the donor register side of the. So what
I
does that
see.
look like? And
Hmm.
could we pick that up in this kind of massive network diagram? Um,
Yeah, because they only have two registers, right? Or two memory registers. There's not like an infinite amount of memory registers
right.
here. So they only ever see pairwise interactions in this analysis.
Yeah. They only see interactions from the last thing that was recombined in that area.
Hmm.
Um,
Hmm. Hmm.
so that's kind of that. I mean, that's a very interesting thing that I feel like could be worthy of more. I mean, this is a great technique, I think, and this is where where the idea of DNA imaging comes in. The idea that you can use these DNA sequences to build up an image of what the bacterial community looks like, and I
But I think they get growth, don't they? They get populations forming, right?
right,
Like back in the example of S5B, right? That D3, that's not a single strain, that's a whole population. So they have three separate populations of D3, some that have interfaced with each of the recipients. And that's going to be the same case in 4C. Although I guess it's not because they prepped it by saying they only have one. They've introduced this concept of like a single cell holding a single right barcode.
right. Um,
But that might not be true, like because things will grow, right? This is a growth dependent writing process. So
Right.
they'll have a generation they'll have a generation, and there are like, basically, yeah, like I see what you're saying. It's not right to think about this as like a single snapshot of everything that the thing has seen, because they only record the pairwise, and that, yeah, like one of these cells could have seen multiple of these donors or recipients, but we'd never know that, right?
Right.
However, as long as that cell replicated, we do have a history of it though, right? That like it has, we know that like, because like the parental cell, I guess, or like some progeny of it has seen them. Yeah, I think I understand what you're saying now as I've talked it out a little bit.
Yeah.
Yeah.
So that's my kind of one little takeaway from that.
The
Um,
resolution is just a bit fuzzier than what it would like. It's not
yeah.
directly, the resolution is not as clear cut as this graph, right? It's not that this thing saw these different things. There's a cycle of growth. It could be that the daughter of that recipient saw the donor, right? The daughter cell of
Right.
that recipient saw the donor. It could be that they all saw it or they all didn't see it. This is also in liquid culture, right?
Uh, I did. Let me check back to see.
I feel like they did this in liquid. So like that is also, like there really is no structure in liquid culture. Or
Yeah.
there's not that much structure in liquid culture.
Yeah. I think that that's right. So you are not so much mapping like locations, but you are mapping relationships.
Yeah.
Uh,
But it might have been interesting to see this more complicated strategy done on solid. Okay, wait, I want to make sure that I'm saying.
yeah. So I'm just gonna because the next figure was quite a difficult one
Okay, yeah, let's
to
let's
get.
move on.
Yeah, next figure is where they measure the number of conjugation partners between donors and recipients based on this thing called the degree of donor barcode or degree of recipient barcode, which is defined as the number of unique interactions that each donor barcode makes with the recipient barcodes, which is equal to the sum of elements in that column that we saw, corresponding to the barcode and the
Yeah,
connectivity matrix. And
this is them trying to get at the information that we're saying it's hard to parse, right?
yes,
Yeah,
that's right.
because if something co-occurred more often with that thing, then it's more likely that that donor has had interact, right, has been in the same spatial location as those other recipients, something like that.
Yeah, definitely. So yeah, what they kind of find is that let me see, because this is interesting, because um, yeah. So I mean, because what I usually think for this, so these should be symmetrical, surely, but they're
Hmm.
not.
Hmm.
So you'd expect is like the donors would like replicate with recipients at the same rate as the donors, but
Yeah,
it's actually quite different. Um, the degree of barcodes for the recipients doesn't seem to be as fitting to that linear kind of thing that we'd usually expect.
yeah.
Um, so yeah, that's all I could make out of this. I
What did
this
what did the authors say about this?
what the let me put my notes. The authors say
They correlated. They suggest that the mixtures.
the weak correlation between the degree of recipient partners suggests other factors such as identities of recipient cell partners might affect the frequency of successful conjugation at the tested donor recipient ratio.
Yeah, actually, it's kind of like, what about the bar? Is there some weird thing going on with different barcodes? That seems odd. It's such a small. They don't make
Yeah,
anything.
I wouldn't. Yeah, that. So I mean, for the donor barcodes, they essentially say that the strong correlation between the degree of donor barcodes in the parallel current conjugation experiments suggests that the transfer of barcodes from donors is not dependent on the identity of their conjugation partners.
Yeah.
So, uh, yeah, we're
Hmm.
having a couple of different ways.
Okay.
Uh, yeah, I'm I'm okay with moving on from this one because I'm
Yeah,
a
I'm okay
bit
with moving
baffled
on.
by
I
it.
guess like but the last thought maybe I want to talk about here is like something about having just the two registers seems like a limitation in this particular setup.
Right,
Like if they
but
had multiple registers, but they couldn't. they couldn't control which register got written to.
Right.
Yeah.
Yeah. So it's going to be a tough thing because what you'd want is basically for it to make a decision at some point. If the register's already been changed, somehow that makes it unlikely to be changed by
Again.
the next
Yes.
again.
Yeah.
So
Some
we
right protection.
yeah, essentially you'd want something like that.
Yeah.
But that's going to be quite challenging to work into there.
Yeah.
Um,
All right. Something about future development in extending this particular tech.
Yeah. Next is using this in continuous evolution experiments. So this is where going back to the phages and using them to inscribe a diverse like kind of sequences into specific areas. In this case, they looked at the lac operon.
Yeah.
Um,
Yeah, so here the
so
diversity of, right, this is very similar to other, right, we've seen continuous evolution before on our shows, and that always it's always about creating diversity, right, and then purifying out from that 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 diverse population, the thing that has the property that you want, and then going again, putting the diversity back in and purifying out the population that you want. And we already saw that they used phage before. They were able to put their constructs into phage, meaning that the phage hold the diversity, can continuously impart that diversity on a growing population of bacteria.
yeah, and this so what they have is a couple of phages, and they have it several stages of like recombination, where these phages have like very diverse like sequences,
Yep.
and then they dilute it and transduce them again to re-add more
Yeah.
more
Sorry, are
diversity.
you looking at 5C right now?
5C,
Yeah, yeah,
yes.
yeah, yeah. Yeah, so yeah, the phage are the ones that have all the diverse sequences. They're all targeting two sections of the lac operon in this case, and
Right.
the selection in this setup is the ability to use lactose better, right?
Yeah.
Yeah, glucose better. And so like if they get more expression of this operon, right, because of the recombination event between the because of the right event between the phage and the bacteria, we'll get bacteria that grow more quickly. And so when they dilute the thing, they dilute the mixture of the bacteria mixed with the phage. They can add more phage or something like that. They'll have a greater population of the bacteria that grew faster, meaning that they got a good phage recombining with them. And then there's the opportunity for those bacteria to meet another phage that adds the next step, right? That makes them even better.
Right.
And so that's sort of what we're seeing in that top row, right? Is that, well, there's an example in the sort of medium gray of one mutation event that made those bacteria grow easier. And then in the next well, they see another phage that gives them another mutation event, right? That allows them to grow even better. And over time, that should enrich for bacteria that have accumulated mutations from these phage that give it that benefit of faster growth.
Yeah. So the next slide should looks at how that process happens in reality. So they've first got them like on glucose, and they do all their mutations, and then they put onto lactose
Yep.
and see which one of these have the right mutations to survive.
Mm-hmm.
So yeah, you got this big peak where everything's fine, and then they switch off to lactose, and very few survive.
Yep.
Um, they're all growing a they're much fewer unique variants. So that basically, so this measures how many variants there are, and this one filters out all the ones that can't survive.
Yep.
And
And
in figure,
this is the selection pressure, right? That should be driving our evolution. And the idea is that, I guess the thing that this H-scribe technology adds into this is that, like, you don't have to change up, I guess, the mutating factor that's going in each time. Like this phage library is constantly imparting some right cycles onto the genome, and as long as you continue cycling this selection, you should be getting better bacteria coming out.
yeah, which brings us to figure 5D, which compares some of these variants to each other in terms of allele frequency, I think.
Mm-hmm.
And we see that some start to develop quite good, start to increase in the growth rate under lack.
Mm-hmm.
Um, yeah, and with and even to the point where some of them outcompete the original wild type.
Yeah. Yeah. That's
Yeah,
what it's kind of so they only show us oh, 'cause yeah, yeah, it's interesting to see that like there is constant change after they've switched over to the lac, right? Because and there are different variants that are coming up in the later days, right? So
yeah,
it means that there are like the phage being mixed in this mixture, like it is helping, right?
yes.
They are doing right cycles and changing those bacteria, and the best bacteria are emerging from the top of all of this.
Yeah,
I I
and
actually think. Oh, I was just gonna say this is a funny model, even just to think about like evolution in general,
yeah.
whether or not like there are phage that perform this function in the environment.
I mean, it wouldn't. I mean, I think we can come up with like a couple of examples of like where phage genes become useful genes for hosts. I mean, I think
Yeah.
there's uh,
But
a
in
lot
this
of.
so but in this example, right, the phage are essentially just a source of
Mm,
right of variation of the bacterial genome. So like thinking
yeah,
like this is essentially like it's like a rare event, right? But like phage that do the transduction of random genome bits, right? Like improperly
yeah.
packaged phage particles. Essentially, what we've made is like a huge library of improperly packaged phage particles that are constantly interacting with our bacterial population, and under a selection pressure, it's giving us a better bacterial population.
And one thing I would like add to that is that if phages in nature when they do this would leave scars on the genome, whereas this one
Yes.
is
Yep.
almost entirely scarless, which
Yes.
I think a really big difference here because usually a
Yep.
phage would inactivate something completely. It'd be a very rare event would just make a very subtle change
Right.
to a genome, and
Great point.
think
Yep.
yeah, I think the subtlety here is like the important part.
Yes.
And I figure S8 is essentially showing the same thing that we've seen before, but in more replicates, because evolution experiments can go all sorts of different ways, I think.
Hmm. Yeah, no, I think that
And
that's the really important point. Again, like a part of trying to understand this paper is like what are the benefits of this system over other tools that we have in our toolbox, right? And this idea of scarless and cis independent, right? Because the other thing about phage is that like actually they usually rely quite heavily on cis elements to get integrated into the genome, right? It's only through the improper packaging of phage particles do you get things that, can recombine anywhere. But even then, it's sort of restricted to like whatever zone was close to those phage genes, right? Getting packaged in. This artificial system is putting all of that in our control, right? Like we choose the things that are going in this phage.
right, and I think that brings us back to Figure One, where we kind of judge how well this has lived up to what it wants to do.
It's promises. So you were saying that, and we sort of like stumbled through, I think, those figures that the spatial, right, like this idea that you can see spatial information, aka cellular connectome, right, between
Yeah,
single cells. Um, kind of you see that, but it's hard to say what's gone on exactly because there's these rounds of replication that have occurred. Then it's hard to like it really complicates matters, right? Because in every round of replication, that sets new daughter cells that have some old inscription on them that then they see new cells. Some subsection of them see the next set of interacting cells, and you get an idea of what's touched each other, but maybe not in what order.
yeah. And I feel like that this is like there's a lot of really impressive things that are going on this paper.
Hmm.
I mean I feel like the rewriting genomes in the community has a lot of potential.
Hmm.
I think the space. I mean, if you're studying conjugation events, then I feel that this model can be very interesting. If you're looking
Hmm.
at, say, how one plasmid can go through a community,
Hmm.
I think that or like how and one of the things that people are trying to study is how communities respond to stress.
Hmm.
And if you, I'm thinking back all the way back to that biofilm paper we did a while ago, where we see like these bacteria like flowing out, and these videos of like, and imagine like how they behave in a certain area of stress.
Hmm.
Spatial information can be quite interesting in that sort of where I mean where like if especially if you're thinking about how biofilms form
Yeah.
and what and trying to capture what happens in the initial conditions of those biofilms forming, which is very difficult to do.
Yes.
But using something like this can be quite an interesting technology to do that.
Yeah, like when we looked at that initial biofilm paper, I think they were using a reporter, right? That like
Yeah,
in a certain condition, like right when a certain condition come on, they would see that reporter go on. And one of the powerful things one of the tools they had at their disposal, which was really useful in figuring out what was going on in that phenomena, was they were real time imaging those initial
yeah,
events, but this technology might be able to sidestep that potentially, right? Because they don't have to image it; they could just see that there are some cells that underwent that particular phase, or maybe they could move into a system where we don't have eyes on it, right? Where we don't have
yeah,
that imaging capability, and now we can ask like, yeah, in this scenario, do we see those little spikes in stress that happen that correlate with different promoters being turned on? Yeah.
so yeah, I think so. I think in summation, I think this paper has quite a lot of potential. There's a lot of really interesting tools they have here and that they've shown.
Yeah. Yeah, I'm sort of
And
excited. Like now that we have some background on it and we're seeing some of the applications, I guess the exciting thing would be to see like where is it used, right? To piece apart certain complicated biological phenomena, right? That's what we're interested in.
yeah, definitely. So yeah, I
Yeah,
think that
because they gave us like they they gave us they gave us this like continuous evolution, like that's like a technology building sort of application,
yeah,
but maybe where our interest kind of lies is like how's it going to be used as a research tool to give us some insight into something like we haven't seen before, and they don't really have a good toy example for that, right? Like
right.
the closest they got was like just showing us that like they can reconstruct like the spot pattern but like we'd like to see it maybe in like a biological system of some relevance, right? To
Yeah,
bacterial lifestyles. Yeah.
yeah, for sure. And obviously, again, they've only done this in E. coli. I would love to see
Mm,
whether this would be
mm,
portable to other bacteria. I mean, if anyone's
yeah.
worried about, I don't think this could be work in like humans or mammalian cells or eukaryotes because the immune system would go crazy, I think, and not
Yeah. But but as I said, like you were sort of in the depths of technological hell there, but like I was saying, like it would be really cool though if they could get it to work in immune systems because like we always think about the immune system as having those pairwise interactions as being really important, right? And wanting to know like what's going on in that, right? And they always talk about like cell fate. There's a series of reporters in immunology,
mm,
right, to determine like the cell fate. Like at some point it was turned on, and then that left that made them GFP positive, right? It's usually like a Kree-Lock system of some sort, and it's like it would be really cool if it could be adapted right into studying immunology because that's
yeah.
exactly where this type of stuff is really important.
I mean, going even like well, going a bit more esoteric. I mean, even like say streptomyces or bacteria where different cells can perform slightly different functions. This can be
Hmm.
quite an in. I mean, well, it all depends on conjugation. That's the only thing that is kind of the limiting factor at this point is
Yeah.
that it relies on that whole imaging is reliant on that conjugation process. If they can find alternative
Hmm.
processes to also ensure that the exchange of DNA can happen. Uh,
Yeah. Well, I was actually thinking like in type 6, right? In contact dependent killing sort of scenarios,
right.
right? Maybe there's a reporter that they could use. So like they would just engineer the whole population to have the H scribe in it, but the H scribe would be responsive to something that is contact dependent.
Right.
Um, yeah, so
Yeah.
like it's not about the transfer of the barcode necessarily, but just like the recording of some sort of transfer. And maybe that's moving
Right.
away a bit from their idea of the barcoding, but like that's just how I would imagine it giving us some interesting cell-cell communication data.
Yeah. I feel like the DNA imaging is very much limited to the conjugation events, but the
Yeah.
DNA writing is very flexible, and
Yes. Yes.
depending on how
Yes.
you build your circuit and your promoter, you can
Yes.
actually do quite a lot with it, I think.
Yeah, yeah, we don't have to focus so much on that DNA barcoding part to get some of the information of cell-cell communication
Yeah.
happening. Yeah.
I just realized I put DNA imaging in the title because it sounded cool, and I had to say something about it.
Yeah. AKA, it was sort of clickbait, and we're actually more interested in the DNA writing part.
Yeah, sorry.
Okay, well, join us next week for our news week. Well, we will survey papers to try to find something that we want to cover in detail the following week.
We want to remind everyone that while we're very enthusiastic about microbiology and somewhat qualified, it is possible we didn't get everything right. Science is about thinking critically and asking the right questions. So if you have any questions or corrections, please let us know in the comments.
I agree. You can reach out to us over Twitter with the hashtag #MicroTWJC. We both believe that peer review is a process and that we can all participate in it. So we hope that you had a good time listening to us ramble about microbiology today. If you think that you have something to add or found something unclear, just let us know.
It's been a pleasure chatting with you, Danny.
Same here, Fuzz.
Bye!
Bye-bye.